Have you ever wondered what gives you enough energy to run around all day? Or what regulates your blood sugar levels to prevent you from developing diabetes? Well, you have proteins to thank. Proteins are essentially extremely complex molecules that build, maintain, and replace the tissues in your body. If we were to look closer at the structure of proteins, we'd realize that they come in all sorts of sizes, shapes, and types. But in the grand scheme of things, they're only a few nanometers big. And each protein has a unique and specific function. Some proteins give cells shape, others act as signals between different parts of your body, and others are required for metabolism to put together and break apart biomolecules needed by your cells. While proteins are some of the most diverse organic molecules, they are all made up of similar components. More specifically, you'll find that all proteins consist of compounds called amino acid residues, which are held together by peptide bonds, which we'll talk about in a second. Amino acids are the building blocks that make up proteins, similar to building blocks of Lego, which you can put together to build a house. The sequence of bases in our DNA determines our unique genetic code and specifies which amino acid will be added next during protein synthesis. Once you put various amino acids together, they form peptide bonds. We can essentially consider these as bonds between amino acids. And then, the proteins begin to take on their 3D shape. This 3D structure determines the function of the protein. For example, collagen proteins in your skin have cord-like shapes, so they can transmit tension between cartilage, bones, and skin. So yes, just like I said at the start of this video, proteins are to thank for helping us gain energy to go about our day, among other things. Let's take a look at a very common and important protein in our body as an example of what they're capable of doing. Cytochrome C is a key protein found in your mitochondria, the powerhouses of the cell, and it helps with the creation of energy for your body through the process of cellular respiration. More specifically, it's involved in the electron transport chain of the mitochondria. The role of cytochrome C is to carry electrons from one group of proteins in the mitochondria to another, so that your body can harvest energy from this electron transfer to make ATP. If you don't understand that, that's okay. All you need to know is cytochrome C is essential to give you the energy to go about your day. Right now, you're looking at the 3D structure of a cytochrome C protein. But have you ever wondered how scientists are able to make models of such tiny and complex molecules? Now you might be thinking, can't we just use DNA to model protein shape? Unfortunately, it's not so simple. DNA tells us the sequence of amino acids that code for your proteins, but what it does not tell us is the protein's shape. This is actually where Leventhal's paradox comes into play. The gist of the protein folding problem is summarized by this paradox, which states that it would take longer than the age of the universe to count all of the possible configurations of a protein before you would reach the true 3D structure. In order to solve the protein folding problem, scientists have been given the daunting task of predicting protein structure. And so, that's where computational protein design, or CPD, comes in. CPD strategies find the best sequences for a protein fold by searching over databases of different amino acid residue side chains and protein backbones. So far, protein design solutions can be categorized into two main categories: ensemble backbone design and flexible backbone design. Ensemble backbone design is when multiple protein backbones composed of a specific genetic sequence are first created. You can think of a protein backbone as what holds a protein together to give it an overall shape. With this method, scientists find the best genetic sequence independently for each protein backbone within the ensemble. You can think of it like a puzzle, where you have one piece and you look through all the other pieces in the puzzle to find something that matches. Except this time, the one piece that you're holding is the protein backbone, and the matching pieces are amino acid sequences. On the other hand, flexible backbone design explores different protein backbones and amino acid combinations at the same time. So now you're changing both puzzle pieces in your hands. This way, you can explore different protein backbones instead of being restricted to just one. Alright, let's walk through the basics of the general computational protein design process. Imagine you have a known protein backbone. This would act as your fixed scaffold. Different amino acids would be modeled at each of the positions in this scaffold, and potential mutations are suggested based on the amino acid interactions within the scaffold and with each other. The main task for computational protein design is to find the best conformation of amino acids for each position so that the protein folds are stabilized and the desired protein function is achieved. Amino acid sequences and the conformations are then scored using a set of energy functions. It's almost like a test where these sequences and conformations are assigned a score based on the quality of the protein that was designed. These energy functions grade the protein so that the computer knows whether the specific shape is good or bad. While the specific scoring guidelines vary for different CPD algorithms, they all serve the same purpose. In fact, there isn't even consensus on a specific set of functions that you have to use just yet. Now, once you combine all the functions together, several optimization algorithms, such as Monte Carlo or genetic algorithms, can be used to solve the design problem. That's basically a fancy way of saying we have mathematical algorithms to help us actually run this code. So, to recap, in computational protein design, firstly, an algorithm creates all sorts of combinations of protein backbones and amino acid sequences. These combinations are then given scores so the algorithm can differentiate which designs are better than others. And finally, the protein design that has the best score is determined to be the final structure. And so, that's how we get structures like this cytochrome C we were talking about earlier. Pretty neat, right? Now, more recently, Google developed an AI algorithm called AlphaFold in order to predict 3D models of proteins based on prior research using large genomic data sets. The model creates shapes from scratch without using previous protein backbones as templates. Simply put, machine learning algorithms are given huge amounts of data, and they try to find patterns in this data in order to make predictions. Thus, this algorithm is able to predict protein structures, which are also scored with another algorithm in order to see how close the predicted structure is to the actual protein. It's a similar idea as last time. Something special about AlphaFold is that it optimizes its scores through gradient descent, which is a mathematical technique used in machine learning to make incremental improvements. I won't bother you with the details of gradient descent, given that it is complicated and requires you to know calculus. But what I will tell you is that they were able to simplify the prediction process while maintaining high levels of accuracy thanks to this technique. As it turns out, the AlphaFold algorithm has been extremely successful, and the code is all open source online, meaning you can check it out yourself. According to the Critical Assessment of Techniques for Protein Structure Prediction, which is a global competition to measure progress on the accuracy of protein shape predictions, AlphaFold was, and I quote, unprecedented progress in the ability of computational methods to predict protein structure. Now that we understand how CPD works and have heard about another really recent yet promising solution to the protein design problem, why is all this important? What's the so what to knowing different protein shapes? There are actually numerous applications of this type of research within biology. On a higher level, knowing protein structures improves our understanding of the human body. It allows scientists to target and design new, more effective cures for diseases. It helps with drug discovery and engineering proteins to perform certain functions. Let's look at some of the examples of the applications of protein design. In 2019, scientists created synthetic proteins that could shrink cancer tumors in mice. These proteins stimulate the same receptors as a powerful immunotherapeutic drug. Insulin has also been the subject of protein engineering efforts to prolong the duration of the action of the drug. For those of you that don't know, insulin is necessary to regulate blood sugar levels in your body. People with diabetes need insulin since they experience either too high or too low levels of blood sugar. In this particular study, an insulin drug called Levemir was modified to allow prolonged releases of insulin into the body, thus further improving the drug to help diabetics. And these are just some of the examples of the many applications of protein design. By combating density-dependent factors like diseases, protein design also could lead to humans living longer lives and, in turn, contribute to population growth. Cancer is the disease which causes the second highest number of deaths in the world, while diabetes is ninth. To think about developing potential cures or treatments to these diseases with protein design is mind-blowing. Above all, I hope this video gave you a sense of how computational science and biology can easily be integrated to solve unique problems. I thought that computer science and biology were completely separate entities for the longest time, but computational protein design is just one example of many of how we can harness the powers of programming and computers to solve some of the major problems in biology. The field of computational biology is truly amazing. With the rise of machine learning, new projects like AlphaFold are beginning to revolutionize this field and bring us one step closer to solving the protein design problem that scientists have been tackling for so long. With much of this research still ongoing, I'm super excited to continue staying up to date with the latest advancements, and I encourage you to do the same. Thanks for watching!